Good morning, everybody. It's 9 a.m. here in San Diego, so we'll begin today's webinar. Today's webinar is on ICM virtual ligand screening. My name is Andrew Ori, and if you have any questions after the webinar, please feel free to get in contact. This is the fourth webinar in our series on from ligand docking to ultra-large library screening. We've covered ligand editing and optimization, ligand docking using fragments or covalent or regular docking. 2 weeks ago, we covered how to prepare chemical libraries for virtual ligand screening. And now in today's webinar, we'll be covering how to perform virtual ligand screening of databases. Future webinars will cover how to incorporate receptor flexibility in ligand docking and screening. And then the last webinar in the series is on ultra-large chemical library screening with ICM. If you have any questions during the webinar, use the chat option or contact us afterwards. You can access all the webinar recordings on our YouTube channel. If you just go to YouTube and search for MolSoft, you'll find it. And all participants in the webinar can request a two-week free trial license of the software. So today's topics will include how to set up the docking project for virtual ligand screening, how to configure different screening parameters and filters, how to execute virtual ligand screening in the graphical user interface or on the command line, how to parallelize jobs on the server or cloud, and how to use job schedulers such as SGE or Slurm, and then how to analyze the results of the screening, looking at the scoring, looking at different analysis tools, and also how to refine your hit list by molecular dynamics. Just a brief introduction to MolSoft, if you're not familiar with MolSoft. We're a San Diego-based company founded in 1994 by Ruben Abigan. Max Totroff is our Chief Technology Officer, and Eugene Rausch is our Principal Developer. Over the years, we've competed in many, we've developed different methods for docking, modeling, and screening, and also competed in many of the comparisons and docking challenges over the years and modeling challenges. More recently, developing methods for not only physics-based approaches, but AI approaches for generative AI modeling and ultra-large virtual screening. The ICM software, which we'll be using in this webinar, runs on Apple, Mac, or Linux. We're just focusing on the virtual ligand screening tools in this webinar, but there's a variety of different tools for drug design and optimization, protein-protein docking, molecular modeling, homology modeling, loop modeling, as well as there's tools for molecular dynamics and Monte Carlo screening. There's also a variety of cheminformatics tools. Some of those we covered a couple of weeks ago in preparation of the databases for virtual screening. There's also bioinformatics tools. You can build alignments and link those alignments to structure in 3D and other tools. So what is virtual screening in ICM? So you would start your screening from different libraries. They could be from hundreds to thousands of chemicals through to trillions, depending on the method you want to use. We have in ICM two different approaches. You can either use a ligand-based approach. So if you have one or more lead chemicals with activity data, you can superimpose those and you can either screen against 3D pharmacophore explicit points in 3D, or shape, or against these clouds are 3D pharmacophores using atomic property fields in ICM. And that method is called RIDE. But today's webinar is going to be focused primarily on structure-based approaches. So you'd have one or more crystal structures, and then you can dock and then score your chemicals to that pocket in the crystal structure. If you're screening millions, hundreds of thousands to millions, you can use ICM-VLS, which is our CPU-based approach. When you get to the trillions and billion size libraries, you would need to use ultra-large screening methods that we have, which we're going to discuss in a couple of weeks' time, Ridge and GigaScreen. So the hit rates are generally around between 20 to 40% for ICM in different projects. The method we use is bias probability Monte Carlo. So when you're using the docking approach, the ligand is fully flexible during docking, and the method has a local gradient minimization to optimize the docked ligand's internal variables using 6 positional variables that are freely and all freely rotatable bonds of the ligand. So we build these inside. You see this purple box. So you take the crystal structure and you build maps, pre-calculated grid maps for hydrogen bonding potential, van der Waals interaction potential, hydrophobic potential, and electrostatic potential. Ligand position and internal torsions are optimized by stochastic Monte Carlo search in ICM. ICM stands for internal coordinate mechanics. We use internal coordinates rather than Cartesian. It's a local gradient minimization step. Ligand is flexible. And then you evaluate the energy in force field, and we place that ligand conformation in a stack, and then we repeat the optimization procedure and comparing different ligand conformations in the pocket and comparing the energy and then ranking them in a stack. And then they're scored. We'll talk about the score in a bit. Just some recent examples of VLS success stories. This is a story from the scientists at Chem Space. They competed in the CASH challenge, which is focused on specific protein targets, and they have different challenges. This is, I think, challenge number 2 that this is on. It's on. So there's currently 6 challenges, I think, at the moment, and they've only published the results of 2 of them. So each competition includes like a hit finding and hit expansion round of prediction and experimental testing. After which all the data is made publicly available. So the target was a challenging target. Is a helicase, SARS-CoV-2 helicase. It's also known as like NSP13, non-structural protein 13. It's considered a promising drug target for COVID therapeutics. And the enzyme plays a critical role in the replication and transcription of the virus by unwinding RNA and DNA. The scientists at ChemSpace, they screened 176 billion chemicals from the enemy in real, and they had hits in the range in this micromolar range. Another success story is from the NCI, Mark Nicholas's lab and Nadia Tarasova's lab. It's just an example of one of a success story. So STAT3 is a transcription factor linked to cell growth, immune response, and also obviously cancer progression. It's often overreactive in cancers and immune disease, making it an attractive target. But the problem is STAT3 it's kind of been labeled as a non-druggable target. So they used ICM to identify pockets in the structure. Most of them are very flat because they're protein-protein interaction sites. but they were able to, by using a combination of our ligand-based virtual screening and structure-based screening, they were able to identify a number of hits to this undruggable, so-called non-druggable target. Just as another example of the validation of the method, a collaboration called D3R, which was a collaboration, an academic and pharma collaboration, where they provided complex co-crystal complexes which weren't published, and the idea was that the software developers were challenged to predict ligand, the docked ligand pose, as well as the ability to discriminate binders from non-binders in a virtual screening scenario, and also prediction of binding energy. And in the three challenges that we competed in, challenge 2, challenge 3, challenge 4, ICM outperformed all the other methods for those three examples. So ligand redocking, screening, and also binding energy prediction. You can read the 3 publications here, which I can make available to you. Just as another example for covalent docking and screening, this independent paper was published comparing AutoDock, CovDoc, Gold, and ICM-Pro and MOE, and they found that the accuracy of ICM was 90% compared to other software. It's an interesting paper because it's completely it's a covalent, although it's a covalent screening. There's also a non-covalent test between ICM and Glide that's reported in this paper as well. So it's worth checking out this paper. Some of these results could have been improved a little bit because there were some crystal-crystal contacts which affected the results a little bit. So we're going to start by going through the steps to set up the docking project. And the docking project is then used for virtual screening. So the way to do this, we're going to use the examples that we've been using all the way through this webinar series, which is the EGFR kinase, and it has PDB code 1M17. So when you open ICM, you will see this window. I'm going to go relatively fast through these steps because most of this is covered in the previous webinars. So we're going to load PDB 1M17. So you just type PDB 1M17 in the PDB search bar here, and this will load the structure. We are going to convert this. You can see that this has allottinib bound, this ligand, this EGFR inhibitor, first generation EGFR inhibitor. And this is the protein structure, and there's some water molecule. We're going to convert this into an ICM object. So you right click on the name, choose convert PDB. We went through most of these options in previous webinars, but we're going to just keep we're going to delete loose waters. So these are waters that aren't making any hydrogen bonds with the ligand or receptor. So they're just sort of floating in the structure. We're going to optimize hydrogens because we're reading it from the PDB. We're going to also optimize asparagine, glutamine, proline, histidine residues. So there's some ambiguities in the crystal in the maps of the crystal structure. So this will optimize the hydrogen bonding network for these, and also the correct protonation states. I'm going to choose to replace the original just for clarity. It's going to overwrite the X-ray structure. It's going to click OK. So we could, to set up, we could run, go to Tools, 3D Predict, ICM Pocket Finder, and this will find pockets within the structure. So we have the ATP pocket here. So we have, this is the N terminal of the kinase. We have the hinge and C terminal, and there's some allosteric sites that could be possibly targeted. And we covered this in another webinar, but we're going to use this pocket to set up our virtual screening. So to do that, we're going to use the docking menu. We go to New Project here, and it asks us for a project name. I'm going to give the project name VLS1. Then it asks you for the directory where you want to save all your docking project files. And I will choose, there's a subdirectory called webinar here. So it's going to save all our docking and screening results to this directory. Next, it's asking us the name of the receptor we want to dock to. This is the ICM syntax, A underscore, and it's 1M17. You can see it in the ICM workspace, and it's also listed here. Next, we need to define the pocket. So the pocket, it says, is this one. It has the code G underscore pocket, and then it's the first pocket it found, which is this blue one in the ATP site. So we can use that here and just choose the option define site around mesh. And you can see now we have green crosses. So this is sort of determining roughly where we're going to dock and where we'll make the maps. You can also see that the selection is here. These are the blue ones are the highlighted residues around the ligand. So once it's selected, we can check the option make receptor maps immediately and just go OK. And the first stage is it has this probe in the middle here. If you rotate, you'll see it a little bit better. But generally, the probe is where the ligand docking is going to start its first initially, but it's a stochastic operation. So it's going to start there, but then jump to a random part in the pocket. You can move it, or generally best practice, generally just to leave it where it is. And then you need to click go at the bottom here in the left-hand side of the GUI. And so this is the critical step. So this is the purple box. This is where the grid potential maps are made. So if you want the ligand to sample a region of the pocket, a certain part of the pocket, then you need to make sure the box is big enough to encompass that site. So you can make the box bigger or smaller by dragging in the corners like this. So this is where our maps will be made. I'm just going to press go again at the bottom here, and then it will make the maps. So once it's made, so basically now the project is set up. So you will see when we in the directory that you chose for your docking project, you have a number of different files. We have this DTB file, it's a text file, and you'll see later that you can edit that file. We have an OB file _rec. This is the receptor object. And then we have the grid potential maps. we have this probe, which is that yellow initial probe, which defines the site of the initial starting point of the docking simulation. And then you get another, and if you're using template docking, which we showed in the, I think in the second webinar, I'll try and put a link up here on the YouTube. You also have another object for the docking template. So we could just go to see those files if we wanted to. So you should all be listed in this directory. So these are the ones I just created today. See how it will have the docking project name and then underscore. This was one I did last night. Also in this directory, I have the SDF file, which we'll look at in a minute. So you can screen basically any SD format file, or if you have smiles, you need to convert the smiles into SD format. You can download libraries from Zinc, which is a wonderful repository of databases, or you can go directly to the vendors like ChemDiff, Cambridge, Enamine. There are also different kind of libraries, such as natural product libraries. If you're doing fragment screening, you can download sort of fragment libraries from these vendors, or I think also from Zinc. And then there's more focused libraries, which some of the vendors provide, such as ligands that are most likely kinase inhibitors or certain family of protein inhibitors, whether they have covalent warheads or have good CNS properties. So that you can also generate your own combinatorial libraries inside ICM, and you can use those as well. At MolSoft's, if you use ICM, you can download MolCart, which is a collection of about 10 million commercially available off-the-shelf chemicals. It's a good starting point for a virtual screening campaign. So we need to set up the database for screening. So in this directory, you'll see that I have a SD file called chembaltarget.sdf. So you don't have to, but for efficiency of virtual screening, it's generally a good idea to convert SD format and so to index your SD format file. You can do that in the graphical user interface using docking tools index database, or you can use on the command line the command make index chemdb, provide the SD file path, provide the the name of the index that you want, tell ICM whether it's a mol or mol2 file, and by the how you want to index it by the ID number. So I'm going to show you how to index in the GUI. So to index in the GUI, we go to docking, go to tools, and then there's an option called index mol2 file. We can browse here for the SD format file. And you can also browse where you want to save the index to, but I'm just going to copy this path and paste it and just put a file type of .inx to show you here. So this is the SD format file we're going to screen, the top one, and the bottom one is the index file. And you go OK, and it will index it. And you'll see in that directory that we have an index file here. So to start the screening, we go to the initially you go to the docking menu and choose Setup Batch Ligand here. And you could choose it, as I mentioned, you can dock directly from SD format file. Go to docking, set up batch from SD format, but we're going to use indexed database. And so you just choose the index file that you're going to dock, this one, and go open and go okay. So, well, basically now we set up the receptor, we set up the database to screen. Let's go back to the slides. So the next thing we need to be careful of are the preferences for the screening. So we'll see in a minute, we have a score, which is our physics-based score. So if you're screening maybe 20 million compounds, you don't really want to save the 3D conformations of every single chemical because many of them have very poor score and you'll just build an extremely large file of results. So it's generally best to add a filter. The default filter for the score is -32. So the lower the score, the better the predicted interaction. But -32 may be an excellent score for some pockets or a sort of reasonable score for others. So it is recommended to do like a small virtual screen of maybe 1,000 chemicals. A good database to choose might be in chemistry, load example table that loads like about 900 drugs, and you can screen those and you can see the distribution of the scores for those 1,000, and then you can extrapolate that to the 10 million, and you'll see how many you will retrieve with a certain specific score. If you don't want to do the full screen, another good approach would be just to, if you have a co-crystal structure, you can just remove the ligand and redock it and see the score of that ligand. And that's a good cutoff for your virtual screening experiment. So to change the score, you go to docking, preferences, and then database scan here. This gives you the so the first option is score threshold. So you could, if you had a very open pocket, maybe a protein-protein interaction site, a score of -32 might be extremely good. So you may want to just lower it maybe a little bit, make it a little bit higher, so you catch a few more ligands and save them. But it depends on your analysis. If it's a very drug-like pocket, sort of buried, then maybe you want to increase this to maybe -40 or whatever the score you obtained through your benchmarking before you start the screening. We also need to be concerned about other preferences as well. So you see in that dialog, we had the score threshold, and then below that, we have MF score is a sort of industry-wide scoring method. We calculate that as well as our physics-based score, but by default, it's set to very high, so no ligands are filtered out based on that score. But you may want to use like basic Lipinski rules to filter out your database. So here, for example, we have ligand size 100 to 500. So it's only going to screen ligands between 100 and 500 molecular weight. It's not going to screen anything lower than 100 or any ligand that has a higher molecular weight above 500. You can also set other Lipinski rules, such as number of hydrogen bond donors, number of acceptors or torsions, and max and min log P. So you can either, as I mentioned a couple of weeks ago, you can either do this before you start the screening, you can prepare your database, or you can just leave the raw database and allow these preferences to kick in to change to filter on the fly as you're screening the database. So that these preferences are in docking preferences. The database scan ones are here, this log P. Other preferences which are important, if you go to docking, preferences general, this gives you this this dialog box. So you can choose to sample race mix centers. You can also sample double bond cis trans. You can relax the covalent geometry, which is an option. If you're docking macrocycles, there's two different options depending on whether you want to pre-sample the ligand beforehand, which is slower. There's options for keeping the carboxyls in your data in the chemicals that you're docking neutral, and also charging groups. So if you've prepared your charges in a different software and you're happy with the SD file, you can just choose none. If you want to use MolSoft's inbuilt pKa model, you go to charge groups and choose the option auto. And this will charge all the ligands on the fly. And you can go okay. And you will see that we just in our directory, the last file to get updated was this DTB file. So you can also change the filters in this file as well. So this is a text file, so just using a VI editor to edit it, just to look at it. So you can see all the different, these are the box dimensions that we use. These are define the coordinates of the ligand initial starting position. It's different weighting. Maximum hydrogen bond donors. We covered that. There's also a variety of other sort of Monte Carlo parameters you can change here. Here we have max log P, and there's also other this has the binding patch. This reports all the ligands in the also all the side chains in your pocket. And for example, here, this is the parameter we just changed, the charge groups, which is set to auto. And if you, for example, here, you can define the SD file that you want to screen and the index. You can put the path to it here if you wanted just to do it all on the command line and not use the GUI. So you would put the path to the SD file and then also here. So yeah, this is the database location you can change. It's just an example. There's other parameters you can change in there as well. So basically now we have all the files we need. If I just do list VLS1 star. These are all the files we have. We're ready to start the screening basically. So you can do that in the docking menu. You go to docking and then run docking VLS batch. And you'll see this this dialog box. It asks for the project name. In our case, it's going to be VLS1. It's going to output a set of answer files, and you can change the suffix of those files, but by default, it's answers. you choose the docking effort. So docking effort is kind of, it's an important parameter. For virtual screening, we generally, for millions of compounds, we would use an effort of 1. This is the length of the simulation. So you have to have some kind of trade-off between you could, if you increase that to 20, it's going to take probably maybe roughly 20 times longer to dock each ligand. But for screening, 1 is usually fine. For small scale docking, you may want to just increase it to 3 is a reasonable number. There's 2 options, one called store alternative conformations. So it's going to report not only the top scoring conformation, but also keep a stack of different conformations of the ligand ranked by energy. There's an option to score all this stack. So if you save the stack, then you can score each pose in the stack. So not only the first one, which is the best energy, but the other ones as well. This will slow down your docking. The majority of users will have a license type of default. But if you have a cluster license, you will have to change the parameter here, which is available in the drop-down box. You can also use the score filter which we set to -32 here. You can also change it here if you want to. And in the GUI, if you have like 8 CPUs, you can split your screening into 8 separate jobs. So it'd be 8 times faster than running with 1 CPU. So those options are in docking, run, docking, VLS batch here. This is this dialog. So we can set the run just in the GUI. You can set the docking running. I'm just going to use 3 and just press OK, and then you'll see that it says 1 background job running, and use 1, and we'll see a progress bar here as well. You see 1 background job. You want to see more information about the job, you can go to Windows, Background Jobs, and this opens this down here. We have the running time, progress. If you right click, you can also see the output. So you can see now in our directory, we have, let's concentrate on the project we're working on. So we see here we have the maps and everything that we created, the DTP file. But we've also got an answers file. This is where it's placing the 3D conformations of the ligands that we're docking, that are scoring within our VLS cutoff range. And it's also outputting a text file called .ou. It's worth looking at that one just to see the progress, and also it will. So it's saying, you can see that it's giving you the information. It's saying it's docking the index file here. Ligands will be converted to 3D from 2D. There's no pre-generation of conformers in this. The sampling effort, which was the effort value, is 1. The ICM score will be evaluated, and protonation states will be charged, will be predicted based on our pKa model at pH 7.4. And then if we go down, we can see it's loading the ligand, it's docking, it's giving other information, as well as the energy values here, and score as well in this line. Okay, so I'm just going to go back to this. I'm just going to terminate. If you want to terminate the job, you can right click on the screening job and just press terminate. And let's get rid of that. So you can also run in the command line as well. So this is useful if you want to do very high throughput screening, parallelize the jobs. So the script we have is called _doc scan. So to run it on the command line, you need to call the ICE on the, it would be like on the Unix command line. You can also do it in Windows with slightly different syntax. So you would call ICM, you call doc scan, you give the project name. In our case, it's VLS1, but in this example, it says doc1. And then you can have a number of different arguments. So you would this one confs, it says only save and score the top 3 confirmations in the stack. This one, the effort, we had 1, but you can increase the effort if you want to. Then there's other input and output parameters that you can choose. You can also manually split the jobs up as well. So you can say, you can run ICM, doc scan, doc1, and then from chemical 1 in my database to 1000. So on one CPU, it's going to doc 1000. Then you can set another job running from 1001 to 2000 on a different CPU and what have you. Alternatively, which is sometimes easier, is you can call ICM, call doc scan, and then use this argument called proc equals, and if it's set here for, say, for 100, so that means it will take your database and divide it up into 100 pieces, into 100 chunks, and parallelize your docking there. So practically in ICM, in the command line, so I have this argument, this is a variable name called ICM home, just for convenience. So all my my ICM executable and doc scan are located in this directory. So I can use this to define this path. So I'm just going to use $ICM home /ICM64, which is my alias for ICM, the executable. I need to tell it ICM where the doc scan is located. So maybe I should move this up a little bit. So we have ICM home again, and then _doc scan. And then we need to tell ICM the name of the docking project. Docking project is VLS1. And then you can use the arguments. So I was going to say effort equals 2.0, for example. That's the minute, sort of kind of the bare minimum. You can also add, you can have you can define the SD file here based on those arguments I showed in the slide. Just press enter, and it will start. It's loading the ICM shell, and now it's started running, and it's putting the index of the output, showing the output here. So we have the same. You see our effort factors 2, same as before. We could have also, I was going to cancel that. We could also put proc equals 8, and that will divide the database into 8 chunks and screen it as well. So those are the options you have. So you can do it in the GUI or on the command line. So if you're running this on like the Amazon cloud or something, then you would need to have some way of doing it on the, you need to use doc scan for that on the command line. So a lot of your maybe if you have a server at your college or company, they will use some kind of job management system. The most common ones are SGE, which is Sun Grid Energy, or Slurm, which is Simple Linux Utility for resource management. And we have a way of automatically generating these scripts for these job schedulers on your server. So you need to call ICM-64. You need to call the doc subscript.icm. You have the queue type. You choose either SGE or Slurm. You give the table, project name, the number of jobs you want, so how many individual jobs, so like 100 jobs, minus sub will automatically submit it. So here we have the example. Go back to the terminal window, choose ICM-64, and then doc sub.icm, and then queue type equals, we'll just do SGE to start with. We're going to doc the Kemble.index that we indexed earlier, the database. And we want to do it with a project called VLS1. And you want to generate 100 SGE jobs. So jobs equals 100. Oh, so, oh yes, so I've got sorry, dollar thing we can't find. Worked earlier. Just 1 second. Yeah, so this is what you should see. I just, for some reason, didn't like VLS1. Maybe I had. That's just an example. So it creates these scripts, the SGE scripts. Because we're doing 100, it's created 100 of them. If you did minus sub, it would automatically submit it for you on automatically. But it creates these individual scripts. So Q sub is the command in SGE to submit a job, and then you have the file that is created and the directory, and then the contents of that file. You have the ICM-64, which I'm using, and then the it's calling doc scan, which it's calling I use different project name here, the input, and but you could also just do it minus sub, and you don't see this. It will automatically send them to your queue, sub them to your queue on your server. So when it's finished, when the docking is finished, or while it's running, you'll see the answers.object. This is the ICM object with the best solutions for each ligand. You'll see a .cnf file, which contains a stack of multiple confirmations, and the output file. So then you need to create a hit list. In GUI, you go to docking, make hit list, and select the answers object. Or on the command line, you can use scan, make hit list. I'm going to show it in the GUI. You go to docking, make hit list, and then you browse for the answers file that we had. So here, If you split it up into multiple jobs, you may see answers 2, 3, 4, 5, 6, 7, 8, however many independent jobs you ran. So how you split it up. So if you want to process multiple files, you can use asterisks here. And so it's going to, I was going to zoom in just quickly to show you. So you have the, you're looking for the object file, and the asterisk is wildcard. So it's going to process all the files with answers 1, 2, 3, or whatever, however many jobs the processes you run it on. You choose whether you want to import the 2D and 3D, or you can just import 2D, or you can just import without the 2D structures. If you've run multiple screens, you can select the best score from multiple answer files. So if you have screened one ligand maybe 3 times, you can choose the best score from each of those 3. If you screen a very large database, you may just want to select the top 100 or 1,000 hits, and import original columns or read in the original columns from the SD file that you're screening. And then you will get a hit list, and I'll show you that. So the hit list is for this one. So you go to docking, make hit list, and then you'll see a table like this. Okay, so you'll make the hit list. You'll have your original _rec, which is the protein object, and then you have the hit list shown here. So you can sort the hit list by score. This is the physics-based score, and the lower the score, the better the predicted interaction. You can single click, and that loads the ligand into the receptor object, shown here. Then you can also calculate properties using chemistry. Calculate properties. You could calculate molecular weight, for example. We could just quickly do that. Go to chemistry, calculate properties, molecular weight, or some other prediction model that you're interested in. And then we can plot the 2 scores. So we have the physics-based score, and then we have what is called RT-CNN score, which is our neural network score, RT-CNN. So it's plotting these 2 scores. I was going to do the size of the plot based on molecular weight and color by some number of flexible bond, number of rotatable bond. So we can, I just want to replot this. Let's do it by score versus RT-CNN score. And I'm just going to color by molecular weight. Okay. So the 2 scores we have are the physics-based score. Is a scoring function which considers various contributions, such as van der Waals interactions, attractive and repulsive forces between atoms, electrostatic interactions, hydrogen bonding, and also desolvation. There's also an entropy penalty. We also have a neural network score, which is a new score that actually outperforms the physics-based score and other scores. It doesn't use any molecular mechanics or physical energy terms. you'll see RTCNN in the hit list. And then all favorable. So RTCNN is trained to distinguish native-like complexes from decoys. Based only like on the geometries of the putative complexes from the PDB. So all favorable or unfavorable interactions are learned and from training examples. And the key distinction between the RTCNN score and the regular physics-based score is that the RTCNN score has a lower penalty for like minor ligand receptor clashes. The lower the RTCNN score, the better. From our benchmarking, we found that we get better enrichment when you combine both the physics-based score, the VLS score, as well as the RTCNN score. So if you choose like a subset here, or do a sum, or choose the best performing one was actually top 20% of the score, top 10% of the RTCNN score, and a combi approach. But you can select maybe here, you can select the ones that have low physics-based score and low RTCNN score. We can then place that into a new, so you can see the selections are here in red and blue here. We can put that into a new hit list if you wanted to. New hit list. And so we have a smaller hit, just choosing a smaller hit list just for speed for this analysis. So when you have a hit list, you see a button here which has a hammer on it. We have our plots here, but there's also a tools option. You can change the visualization of you see if you single click, it loads the ligand here. You just choose pretty visualization. We have the ligand docked here. If you want to compare a pose, you can use the boxes in the L column to temporarily sort of overlay the docked ligand, the two ligands. We have two here and here. If we were interested in doing more analysis, there's a variety of different tools in this descriptor calculator shown here. So you can, if we were interested in all the ligands, if the ligands in our hit list made a hydrogen bond, you can see a hydrogen bond with this atom here. You can select that atom using the pick atom tool button here and just select that oxygen. And then you choose the option here, hydrogen bond to selection. So it's going to flag all the chemicals that make a hydrogen bond to this atom. It's going to give it a name, column name. So say H bond O, calculate property. And now we can see we should have a column here called H bond O. We can color our plot here by whether it makes a H bond or not, or the energy of the H bond shown here. So I think purple is 0. These ones aren't making hydrogen bonds, but you can sort the column by clicking on the column header. You can sort the results here. So these are all making hydrogen bonds, and 0 means it doesn't have one. You may want to see how your docked ligand compares to a co-crystal structure in terms of the 3D pharmacophoric properties. So if we read in 1M17 again, the structure, it has allotinib bound, the green ligand. I'm going to convert this to an object, ICM object again very quickly. And then we just for the example, we're going to compare how well the ligands that we docked match the 3D pharmacophoric properties of this co-crystal structure ligand. So we just select the ligand, or you could have a substructure. You could cut this down to a smaller thing, and you go to tools, choose APF pose. APF stands for atomic property field. pose similarity to this ligand. And just check here. I'm going to give it a column name APF score. Calculate property. And we should get a new column called APF score. So like the docking score, the lower the score, this is our APF with atomic property fields. So how the lower this score, the better the ligand matches the ligand that we docked, matches the ligand that we're comparing it to in terms of the pharmacophoric properties. You could then change this to color by APF score or some other parameter here. Maybe make new selections. So other tools for analysis are here. So you can also get an interaction fingerprint. You can get interaction on list. If you just click here, and then calculate property, it's going to tell you all the interactions between the ligand and the receptor in a table form. So each residue in the pocket, whether it's making apolar interaction, hydrogen bond donor, 0 means no, 1 means yes, for each of the residues in the pocket. You can also cluster in the tools section by atomic property fields. So this will cluster your hit list by 3D pharmacophoric properties. And you get this tree. So you could select maybe this branch. I'm just going to undisplay some of these things. So if we select this branch, for example, these two, use command to display them, color them a particular color, blue. If we go down further down the tree, we probably find ligands with different, slightly different, or different pharmacophoric properties. So not overlaid. So we select those, and we can display them. So it's a good way of selecting diverse. You can use this tree to select a diverse set of compounds. Or you can select a group. If you have like one hit, you can go back to this tree and select additional similar scoring ligands. Or you can use the tree to select a diverse set by right-clicking and choosing select centers by. So you can say select a center from each branch on your tree based on minimum score, physics-based score, and it will make a selection. You could use this to choose a diverse selection of chemicals from your hit list for experimental testing. You can see the selections made here. The selections propagated to here. You can right-click and then choose copy to new table. Called pick or test or something like that, and you get a new table. It's just I need to remove the selection. Just remove the selection. Go back here. Select centers by, and score. And then you can copy to and copy them to a new table. Got these, so you could maybe save this as a SD file and share it with your colleagues, or if you want to save this all the docking projects in one project, you need to right-click on the hit list and choose a standalone hit list. You'll see this is already a standalone hit list, but you'll see create standalone hit list, and then you can share. You just save the project file, save as, and it will be displayed. So just to end the webinar, These are the hit analysis tools we showed. You can compare APF post similarity, you can do hydrogen bonding, you can go through your hit list and calculate the charges, buried molecular surface, volume change upon binding, the interaction fingerprints, and pharmacophore MSD. You can also, and generally from our virtual screening work we do here for services, we usually stop at that point and we send, a hit list ranked by score. But we recently implemented OpenMM MD simulations and a more rigorous analysis of the hit list. You can run an MD simulation on each of the chemicals in your spreadsheet, in your hit list. So you can only do this on the command line at the moment. You call ICM, you call run MD open mm.icm, and then the time, the different parameters. You provide the hit list as ICB format, so file, save as, and then the receptor object you'll have in your docking project. And then it gives you statistics on the RMSD and the RT-CNN score drift. So ideally you'd see a stable VLS hit would be one that doesn't have a lot of drift. So this example has a variety of different states, so maybe it's not an ideal candidate for testing. This one is stable. You'll see these options. This is all done using OpenMM in the ICM interface. This script is in /bin in your ICM directory. So I just wanted to thank you very much for your attention, and if you have any questions, please feel free to use the Q&A in the Zoom, or feel free to email me afterwards, and we can also send you a trial license as well.